Thanks a lot for the organizers for having me here, and most importantly, thank you so much for supporting the 3 folks from my lab who were able to come here and enjoy this great meeting. So I'm going to kind of stick to what I was told to do or suggested, that is talk about transporters that are targeted to the plastid and discuss what we learned about looking at their evolutionary history and how that might feed back into our understanding of endosymbiosis. I'm going to start with something that has nothing to do with it, but I want to sort of lay the framework for what happens when we have strong selection on cells. This is work that was done by Fatima Faflunker, a PhD student in my lab who's in the audience. Then I'm going to talk about our analysis of some Arabidopsis transporters, talk about the plastidic phosphate transporter translocator family, and then actually talk a little bit about Pollinella, I think a very important organism that hasn't been discussed yet and I think should be something that would be fun for people to think about. So we want to look back a billion years of evolution or hundreds of millions of years. What happens if we look back just a few million years at an alga that's under strong selection to adapt? And the one I'm going to talk about here is called Picochlorum, and it was isolated by Brian Palaniuk at Scripps Institute in San Diego, and Fatima and Shui Wang, his former student, worked on it. And what's interesting about this organism is that it can withstand incredible salinity fluctuations. It can go from distilled water to 1.6 molar sodium chloride after a couple of days, grow beautifully. Really, really interesting organism, interesting to the biofuel and biotech industry, which is why we're trying to develop this, but also interesting to see what alga can really do when under stress. And so what's special about Picochlorum is that it has a really small genome. It's about 13 megabases. It's one of the smallest known. It has about 7,000 genes. And it fits right into the green algae and so on. So we have an interesting model. It's almost a bacterial type chromosomes with genes packed end to end. Some genes that involve in the same function are side by side. So what happens when that cell is under strong selection to survive really horrible conditions like very, very strong fluctuations in salinity? Well, what Fatima found is really surprising. Even under this selection for genome reduction, it has 24 bacterial-derived horizontal gene transfers. And these are not shared with other green algae or plants. They're actually only found in Picochlorum. There's no evidence of bacterial contamination. It's a pretty good assembly. These genes are surrounded by other green algal genes, eukaryotic gene structure, and so on. So we believe they're not coming from contaminants. And guess what they do? A lot of them are involved in stress tolerance. They help provide osmolytes, make the cell wall stronger, get sulfate from the environment, control the cell cycle. This is not proven, but it's really amazing. Green alga appears to harvest cells from the bacterial world and contribute to its ability to live in stressful environments. On top of that, what she found is enrichment of sugar transporters, which is why I'm giving the story, because it'll come back to what I'm going to talk about ultimately. And what she found is that those sugar transporters, she identified them here, some hexose transporters, they actually allow it to grow faster, allow it to withstand high salt stress. The more glucose you put in, the better it does. So that's also really cool, so it's a clear mixotroph. And one of those genes that's really famous is this thing called HUP1, which is only found in these greens, some fungi, and some bacteria. So really sort of a sporadic distribution, but a really well-known glucose transporter that contributes to this alga's ability to live in a hostile environment. So why did I say that story? Because in the short term, looking at Picochlorum within a mesophile that became adapted to these harsh environments, it actually recruited genes from the environment, and we believe that they're part of a toolkit that allows it to withstand these salt changes in the salt concentration. It also has a transporter with a sporadic phylogenetic history that supports mixotrophy. So a lot can happen in a short period of time when enough selection is placed on a cell that even under genome reduction pressure actually is able to do something very special. So that's the prelude, the preamble. Now, what happens when we turn to the archaeplastid endosymbiosis, which is the 1 or 1.5 billion year old thing? So people have thought about how do you figure this out, and lots of people in this room have done this for many years. The parts of this puzzle, when you try to figure out how endosymbiosis went down, that are in decline, are the genomes. Those are the parts that are getting more and more complex that we look at them. The parts that are kind of exciting are the machines, the translocons for protein import. And what I'll talk about today are the translocators that control metabolite movement between the cytosol and the plastid. So we think that, yeah, these kind of machines were probably very important for establishing and maintaining the endosymbiosis. What can we learn if we look into their history? And I worked with Andreas Weber at Heinrich-Heine Universität in Düsseldorf for a long time, and he's worked a lot on transporters, and I think all of you know it, but here's a piece of text directly from his paper, that they're transporters that line the inner membrane of the plastid, have hydrophobic membrane-spanning regions. They move metabolites between the cytosol and the plastid, and they contain a transit peptide that can be used to predict them, but which targets them to the plastid inner membrane. So that's what they are, as I'm sure most of you know. So if you want to figure out something as difficult as where did the archaeoplastid or plastid targeted transporters come from, where would you look? So there's about 150 or 200 predicted, but based on bioinformatics and looking for the targeting sequence and the transporter annotation, but really there's only about 34 to 37 that are validated with physiological data. And this is from Karsten Fischer's review, and I'm just showing him here. Here are the transporters that we know transport what they say they're supposed to transport, and we know that they're localized to the plastid inner membrane. And so this is a good set, a training set, if you will, to figure out where transporters have come from in Archaeoplastida. When we do that, we looked at it in a couple of ways. I've been working with Eric Baptiste at UPMC in Paris now for a couple of years, and a postdoc in our lab who came from Eric, Slim, just did these analyses of which I want to present to you. And when we looked at these data, we thought, how do we get an overall picture of how the transporters are related to each other, which ones are distinct, which ones might share similarity. We used this class of methods that Eric is the maestro of, which is protein similarity, where you generate all versus all blast search, and you connect proteins when they fulfill some cutoff, blast, coverage, and so on, and you generate this overall view of the data. These things are called connected components, gene families, and you can sort of toggle how big the gene families get by making the stringency very high, so they get a lot of small gene families, or make the stringency low, and the gene families grow in size. So that's what they do, and what we did is we took those Arabidopsis validated transporters, we blasted them into a database, and put all the proteins together, about 2,330, and we built a network from them to see what is the overall history of those transporters. And they were built with two constraints, having at least 70% query length coverage in every pairwise analysis, and a pretty low e-value, just to see how far in time we can go, and what does the data tell us. It's a hypothesis-generating step, but it's really fun. Right, and then we clustered local sequences into communities using this well-established Louvain method. Okay, so this is what the Arabidopsis transporter network looks like. Each of these is a gene family. The annotations are shown here. And where they come from are shown here. The prokaryotes are in blue dots. So these are communities where we have prokaryotic sequences. And so when we see that, using those cutoffs that we did, we actually pick up a whole series of gene families that define the evolutionary relationships of the Arabidopsis transporters. And one thing we can see right off is that some of these families, 1, 2, 3, 4, 5, 6, 7, are actually only eukaryotic. That in our database, no prokaryote fulfilled those criteria. Therefore, these look like eukaryote-derived transporters, and 2 of these are plant-specific, if you look at the colors. And the others have a combination of different prokaryotes and so on. So we have 34 families of transporters, 16 large connected components, exclusively eukaryotic, 17 transporters sitting in 7 components. So we can say right off with this broad overview that a large proportion of those transporters are actually appear to be derived from within eukaryotes. What I'll talk about a little bit here is this one, which combines a couple of families together. And here's what that looks like in detail when we annotate the communities based on their phyletic origin. It's really nice. Here there was a couple of copper transporters that were identified in Arabidopsis, HMA1 and PA1. It turns out that they're derived from bacteria, with the 2 Arabidopsis proteins shown on either end with prokaryotes in the middle. And so this suggests that these two copper transporters involved plastocyanin and copper zinc superoxide dismutase in the plastid actually share a single origin from a bacterial source. And this connection to this eukaryotic specific thing looks like it's based on a couple of domains that are showing some similarity. So it looks like this is not a real, that this connection can be cut quite easily if you were to increase the blast threshold. So what does that tree look like? Now we have a collection of two copper transporters with some distributions. If we take all the data and we make a tree out of it, we actually get a lot of bacteria, but we get two clades. The PA1 clade, here's the Arabidopsis with a cyanobacterial origin. We get the HMA1 clade here with a chlamydial bacterial origin up here. So we're able to connect these two together. Although the networks don't tell us what the structure within that data might be, the phylogenetics suggests that these two proteins embedded in the plastic inner membrane, import copper, appear to be derived from two distinct bacterial phyla. Okay, so you can go back to the good old-fashioned phylogenetics, RAXML trees, and so on, and you can see how many of those transporters are of host origin, cyanobacterial, other bacterial, unclear, and so on. And it looks like about half or more of them are eukaryotic derived on a gene-by-gene basis. And if you look at those, here are the 9 clades. This is the total number of transporters. If you look at the actual families of transporters together, you see that there are about 9 clades that are eukaryotic with 2 plant-specific, about 8 clades distributed amongst prokaryotes, and a couple that we don't know where they come from because the trees can't be interpreted very easily at all. So, looking at a set of well-understood transporters from Arabidopsis using a couple of different methods provides an overview of the data, and it's consistent with the idea that a significant contribution from these plastic transporters came from the eukaryotic host, with cyanobacteria providing a smaller contribution. So about 11 distinct gene families are of eukaryotic origin, whereas 8 are derived from different bacterial groups. Okay, so we can focus in now on a sort of an interesting family of transporters. These are the nucleotide sugar transporters, the NSTs. And this is a really interesting one because when we were thinking about this transporter with Andreas a few years back, and this is Fabio Facinelli in his lab who did some of the work I'll talk about, we imagined that the first sugar transporters may have been derived from existing ER or Golgi-derived transporters, that's the NST family, and that by the fusion of the ER with a potential endosymbiont, it could have delivered those transporters to the inner membrane, opening up the connection for sugar transport. Something that Patrick has talked about is something that we wanted to test using phylogenetics. So this work was helped by us getting a draft assembly of the Cyanophora genome, which then we had genome data from the green lineage, the red lineage, and the glaucophyte lineage. We have now a better genome assembly of Cyanophora, which we're hoping to publish soon. And so that allowed us to look across the Archaeplastida with some confidence. And what we found is we could actually place all of those sugar transporters of this phosphate antiporter family as a monophyletic clade with the red and the green showing the red algae and the green algae in plants, and showing that the entire chromalvulate or SAR plus rhizarian clade was all, SAR clade and haptophytes, cryptophytes are all derived in a monophyletic group from within this existing clade. And the nucleotide sugar transporters appear to be sister. That suggests that a gene was duplicated that gave rise to further duplications that then resulted in this complex family of sugar phosphate antiporters that are found in plants and algae. With this happening presumably in Archaeplastida ancestor, and this origin that unites the transporters in this secondary derived clade with another origin in Rhizaria from a green source. So what was missing from that tree was the glaucophytes. In fact, we found glaucophyte transporters, but they all fell in the NST family. Not a single glaucophyte transporter could be found in that phosphate antiporter family, which is where we expected to find it. When we looked at the genome, in fact, we found some candidates for sugar transport, but they were a member of a bacterial UHPC-type hexose phosphate transporter. And here's a tree of the Archaeoplastida showing that they share a common branch with this bacterial UHPC gene, and that the three members of the Archaeoplastida are sitting there, suggesting that the common ancestor gained this hexose phosphate transporter in the branch that unites them. We also looked at the targeting. You can't see that, but I'll point it out in a second. We looked at the putative targeting of those transporters, see if we could find evidence that it could be targeted to the plastid. So what we found for the two Cyanophora transporters was an N-terminal extension carrying a conserved phenylalanine that is a conserved part of the targeting system in glaucophytes. But surprisingly, with the completion of the Galleria genome project and good gene models, we also found evidence that this family, the reds, and maybe even the greens that contain the entire TPT and other phosphate antiporters, also appear to have some targeting sequences that might also suggest that they're also plastid targeted. This is work that's underway in Andreas's lab to test that hypothesis. So the test of the idea that Cyanophora was carrying what appeared to be an ancient version of sugar transport that is from a bacterial origin and was excluded from the red and green gene duplication club was provided by Fabio's work where in Andreas' lab, where they generated a proteome of the inner membrane of the Cyanophora plastid and of plastid proteins, and of 510 distinct proteins they found, there was no evidence of any of the triose phosphate transporters, but there was evidence for both of those UHPC proteins I just spoke about. So they were in fact being targeted to the inner membrane of Cyanophora. To validate that, Fabio made YFP fusion constructs where they took the 159 amino acid transit peptide, showed that it could target YFP to the chloroplast of Nicotiana, and that the entire protein, when expressed and transformed in these cells, also made these sort of crescent-shaped sort of fluorescence that suggested, and is consistent with the plastid membrane targeting. So this suggested that they not only carry the targeting signal, but they're actually localized to the membranes that we expect to find them at. So the work with the Galleria UHPC is underway. The only work that's known until now from Andreas is that it doesn't seem to have a lot of glucose 6-phosphate transport in reconstituted red algal plastid membranes. However, it can suppress a glucose 6-phosphate deficient growth phenotype in E. coli. I'm hoping by the time that we write the paper for December 15th, we're going to have these results in place and can include it there. Okay, so in the last bit of my talk, I'm going to talk about Pollinella. And this is like a lot of great ideas. It sounds good when you say it, and it turns out to be quite complicated. And I'll show you what I mean by that. So Pollinella is really, really interesting for symbiosis research because it is not this. This is the famous cyanobacterial capture and origin of the Archaeoplastida, 1.5 to 1 billion years ago, one that everybody knows and talks about. The Pollinella one is maybe about 60 million years ago, and it's in a single branch of the Rhizaria, thecate amoebae that have a cyanobacterial chromatophore inside. And first described by Robert Lauterborn in 1895, is this a good model for studying symbiosis and in particular primary endosymbiosis? So for a long time it was thought that Pollinella, because there was only one culture that Michael Melkonian had, and then there was one that Ken Ishida had in Japan, that was very rare and it's some sort of a relic species. In fact, my former postdoc in my lab, Hwan Soo Yoon, who went on a worldwide tour looking for Pollinellas, has found them in all kinds of places. So here are the photosynthetic Pollinella. They're found in New Zealand, Japan, a number of places. So they're not very frequent for sure. They're like, shaded, sort of acid bog type water, but they are actually all over the place. As important, we have a large collection of single cell samples for the heterotrophic sister to the photosynthetic Pollinella, which is a great place to look to see if we can find support for the shopping bag model idea of gene transfer that might predate a primary endosymbiosis. Okay, so Pollinella has its chromatophore genome. It's derived from the alpha cyanobacteria. Here it is, that clade. Nothing to do with the plastid that's found in Archaeoplastida and all derived secondary and tertiary plastids over here. So it's clearly a separate event from the Archaeoplastida event. Now here's some pretty pictures. Please note that the strain I'll talk about in a second, the German strain, is 20 microns. The one we have also in culture from Japan is 10 microns. Those numbers will mean something in a minute. Okay, so what has happened in the Pollinella chromatophore genome, that is, that captured endosymbiont genome? Well, here are the sequences for those two Pollinella strain species. So they're very highly conserved in gene order and gene composition. And if you look at, compare the two genomes, we can find differential gene losses. So we can actually show that since they speciated, that we had about 39 gene losses that are specific to the Japanese strain and 27 that are specific to the German strain. So we believe it started with something like maybe a 2 million base pair genome. It's down to about 1 million base pairs. So at least half and maybe up to 2 million base pairs of DNA were lost during the process of genome reduction. So what was lost from that genome? Eva Novak made a great analysis of this, and we followed up later on. This is the predicted proteome of the chromatophore, and the things that are lost are shown in red. They include the TCA cycle, number of amino acid biosynthetic pathway enzymes, and a number of important transporters that would line the membrane of the chromatophore. And these transporters are ABC transporters for polar amino acids, some metals, iron and nitrogen compounds. So we have a genome that's highly reduced from maybe 2 to 3 million base pairs to 1 million. Then we also have really important pathways lost that would take away the autonomy of that captured cell. So we decided that we need to get a much better look into the genome of Pollinella, and we've been trying this for a while. This is a collaboration with Eva Novak, who's now in Düsseldorf, Arthur Grossman at Stanford, and Dana Price, who's here, is a bioinformatics specialist in our lab. And this whole project to generate the Pollinella genome is based on one great thing that Eva did while she was in Arthur's lab. She was able to get an azenic culture of Pollinella. This field has been really sort of stopped and been really, the greatest problem in pushing this organism forward is that we can find lots of species in nature, we can't grow them azenically, and they have lots of bacteria inside of the cells, a lot of Coxiella, other bacteria, so the genome data is always very, very difficult to analyze. So this problem was solved in most part by Eva using this technique, and after many of these transfers, she was able to get what appeared to be a bacteria-free culture of the German strain CCAC 0185. This is the 20 micron cell. So what does that genome look like? That's the obvious question. There's lots of things you can ask about it, endosomatic gene transfer, things that we've been asking for a long time. Well, it's not what we expected. It turns out that if you look at the genome assembly, over 100 billion base pairs of data were generated with HiSeq and MiSeq, assembled, identified contigs that contain 77 core eukaryotic genes. There's a program called Segma with a database where you can then find about 458 genes. The ones that had the Segma genes on them and were complete, we mapped the reads back to them to see what depth of coverage we had. And of course, the 100 billion base pairs of data, we expected to have huge coverage. Well, we had about 10x coverage. So the genome looks like it's probably 10 billion base pairs in size. And so this is a huge problem for us. And we've looked at the K-mer frequencies, we've looked at a lot of things trying to make sense of it, but that's a big problem. So we're working on trying to get a better handle on how to assemble this and whether we need to have long read data to be able to actually do a good job with it. So it's 3 times the human genome size. Well, what do we have? Well, we have about 32 million reads of RNA-seq data from a clean culture of Pollinella. And if you do the same core eukaryotic gene search, we get most of those eukaryotic core proteins, suggesting that we have a lot of contigs, but within those contigs, we have most of the eukaryotic genome sitting there. So that's a good resource for asking questions. So today, I'm going to just talk about 2 questions we asked. The first one is, how many of these nucleotide sugar family members are in Pollinella? Could these have the potential to be retargeted and open that endosymbiont for a connection to the host? And what was the fate of those transporters that I mentioned that were lost from the chromatophore? Did they end up in the nucleus or were they lost? The archaeoplastid example suggests that most of them may have been lost outright. For the first part, here is that same old nucleotide sugar transporter tree. None of them go here, obviously. This is that special plastic targeted set. They all go over here within the expected eukaryotic clade of endomembrane and Golgi-associated transporters. And here are the Pollinella guys. Are any of these transporters being potentially targeted to the inner membrane of the chromatophore? We don't know, but Ava has started a proteome analysis of the chromatophore, and this will be a really, really exciting question to ask. Do any of these guys actually end up there, as we suggest may have happened for the archaeoplastida? It's a testable hypothesis. And why is that a testable hypothesis? Because there is evidence that Pollinella actually has some sort of a trafficking system to the chromatophore. This paper, published in 2012, showed that 3 photosystem 1 proteins encoded in the nucleus were actually, they were translated in the cytosol, and they could be localized to the chromatophore. On top of that, the protein could be localized to Golgi, suggesting that there may be a secretory pathway in place in Pollinella to move these nuclear encoded proteins into the chromatophore with no evidence of any targeting sequences within those nuclear encoded proteins. So there is some reason to believe that there may in fact be some hidden gold if we look at the proteome of this organism. So what about this other question? Where did those transporters go that were on the chromatophore genome? We did a reciprocal blast analysis using the transporter database and the bottom line is we found about 438 high confidence transporters in our EST data, 300 eukaryotic, about a dozen prokaryotic, 3 cyanobacterial origin, none of them derived from the missing genes in the chromatophore. So it doesn't look like it until now we can find any evidence that those transporters that were once encoded in the free-living cyanobacterial ancestor have moved to the nucleus. So my final thing is Pollinella mixotrophic? It's been hard to grow. It's been raised on Varus H, which has soil extract in it. Just for fun, we looked at the transporter data and found a whole number of a series of sugar transporters, amino acid transporters. We don't know what they do. We don't know where they're localized. But here's a tree of one of those. It's a monosaccharide transporter. It may be interesting to think about now adding some organic carbon sources to the Asiatic culture and see if we can get a better growth of Pollinella. Could in fact be a mixotroph. We know it can't live without its organelle, its chromatophore, but it might actually be able to live better if we give it some organic carbon. All right, to end my talk, so you don't get tired of standing there, there's a big caveat here. Bioinformatic predictions of transporter targeting and subject specificity are preliminary. I'm telling you a just so story for the most part. We need to have validation, proteome data, and so on. The comparative data suggests that archaeoplastid target transporters are comprised mostly of host-derived proteins that were retargeted to the organelle. Clearly some very important contributions from bacteria, including the nucleotide transporters, copper and dicarboxylate, and so on. The phosphate antiporter sugar transporters are a great example, and they suggest that if retargeting does occur, that might drive gene duplications and the fixation of a gene family that then becomes destined for that function. Pollinella is a useful model because it has some of the hallmarks of the process of organogenesis. It's permanent, has genome reduction, it has some form of import system, but differs greatly from the archaeoplastida, which I think is a great opportunity to learn something new that we may not have learned from the archaeoplastid example. And the chromatophore proteome work underway in Ava's lab and the complete genome work underway in our lab will help us understand better how gene transfer, endosymbiotic and horizontal, has impacted organelle, the cell, and for this talk, the transporters and what they're telling us about the cell's biology. I want to stop there. I want to thank the folks in my lab. I want to thank our funding sources. And Juan, Dana, and Fatima came with me. I'm really happy they're here, and thank you for your attention.